The coccus, when found in pairs, it is known as diplococcus. When it exists in chains, then it is known as streptococcus. The example for streptococcus is Streptococcus pyogenes bacteria, the bacteria which causes cold and cough to us. And then sometimes the coccus, they exist in groups, in clusters, they are known as Staphylococcus bacteria. Then another type of bacteria, depending upon the shape, is bacillus, which are rod-shaped bacteria. So you know the bacillus found in curd, that is lactobacillus bacteria. You can see here the bacillus anthracis, which is a chain type of bacillus which causes anthrax disease in cattle. Then lactobacillus bacteria present in curd is a bacillus which exists in singles. Okay, now the next shape of bacteria is the spiral bacteria. The spiral bacteria can be divided into 3 different categories. Some scientists, they consider the first category, which I am going to tell you just now, of a spiral, as the 4th kind of bacteria, that is Vibrio. Vibrio are the bacteria which are comma shaped. You can see on the slide the Vibrio. How does it appear? It appears like a comma. The example of Vibrio is Vibrio cholerae bacteria, which causes cholera disease to us. Then the 4th shape of the bacteria is the spiral bacteria, which are mainly of 2 categories. The first one is a spirilla. You can see here Helicobacter pylori bacteria, the example of a spirilla, which is actually found in the pyloric stomach, which causes acidity problem to us. The last one is a spirochete. Spirochete are again spiral bacteria. The example for it is Treponema pallidum, which is very, very important for you to know, because this is a bacteria which causes a type of reproductive tract infection to us. Okay. Now let's see the structure of a prokaryotic cell. You can see, children, there is a bacterial cell drawn here, and very strange, such a tiny cell whose size is just 2 to 3 microns. This tiny cell has got great protection around it. It has got 3 protective layers around itself. The first and the outermost protective layer of a bacterial cell is known as glycocalyx. So it is the outermost layer in a bacterial cell, which can be of 2 kinds. In some bacteria, it is in form of a loose sheath, and that loose sheath is known as slimy layer because it is slippery in nature. And why is it slippery in nature? It is slippery in nature due to the presence of a polysaccharide, a sugar. Okay, then the other category of bacteria which have glycocalyx, not made up of slimy layer, rather their glycocalyx is a tough resistant layer which is known as capsule. The next protective layer around a bacterial cell, or the next cell envelope of a bacterial cell, is cell wall, which is found even in the plant cells also. But here the cell wall, it is not made up of cellulose as it is made up in the plant cells. Here it is made up of a very different type of a chemical known as peptidoglycan. The name itself is telling it has got protein component as well as sugar component. Peptido means protein component, glycan means sugar component in it. So peptidoglycan, they are of 2 kinds which are found in the cell wall of a bacterial cell. One is N-acetylglucosamine and N-acetylmuramic acid. So the chemical nature of the peptidoglycan is that it has got N-acetylglucosamine and N-acetylmuramic acid, which are present in a regular sequence. And this cell wall, it is responsible to provide rigidity to the bacterial cell. Now the last but the most important and the innermost layer of a bacterial cell in its cell envelope is the plasma membrane. It is the membrane which is made up of lipid and protein bilayer. It has got a bilayer, double layer of lipids. In between the lipid layer, there are proteins also present. And what's the function and what's the nature of plasma membrane? Plasma membrane is a semi-permeable or a selectively permeable membrane which allows only selected things to move in and move out of the bacterial cell. So let's see. Now the bacteria is again divided into two categories depending upon its staining property. An organism, how can its cell be stained? So depending upon that staining property, there are two types of bacteria. There are gram-positive bacteria and gram-negative bacteria. Now what is this gram and what is this gram stain? So gram stain or gram staining was the method of staining bacteria which was given by a scientist known as Gram. Okay, so all the bacteria which retain the gram staining even after trying to decolorize it with the alcohol, those bacteria, they are known as gram-positive bacteria. So let me repeat once more. The gram-positive bacteria are those bacteria which remain their stain even after getting cleaned by alcohol. So such bacteria are gram-positive and the others are gram-negative. So let us see how do we go for gram staining. So when we are staining the bacteria, we put the bacterial cell into crystal violet, the stain. And this crystal violet, once it is put, then we add a fixative. The fixative of the stain is iodine. So now the stained bacteria, it is transferred to iodine medium where the color of the bacteria is fixed. Then we will add a decolorizing agent which is alcohol. After adding alcohol, there are a few bacteria which retain their stain still after that. Such bacteria are known as gram-positive, and the ones which get decolorized after treating with alcohol, those bacteria are known as gram-negative bacteria. You can see on the slide. Now we are done with the three cell envelopes which are present around a bacterial cell. Now let us see the processes or the structures which come out from the bacterial body or bacterial cell. So those structures, they are known as cell processes or cell projections. So the first cell projection which is found over the surface of bacterial cell is a flagella. What is a flagella? What is its function? The flagella are the locomotory processes arising from the basal granule. The flagella, it arises from a structure known as basal granule. You can see here in the figure given on the slide. The basal granule is that yellow structure having some 3-4 rings you can see, and emerging out from that is that long process known as flagella. Now flagella has got 3 parts. The first part is the basal granule from where the flagella arises. The second part is that red structure which is known as hook, which connects the basal granule with the filament of the flagella, which is the last part of flagella. Then the hook of a gram-positive bacteria has got 2 rings, while the gram-negative bacteria, they have got 4 rings in their basal granule. Now the last part is the filament. The filament of a bacterial cell is made up of microtubules. Then the next process which emerges out from the bacterial cell surface, you can see in the diagram that blue bacterial cell, it has got certain hair-like projections coming out which are known as pili. Now what's the function of pili? Pili actually facilitates the transfer of genetic material from one bacteria to another bacteria while the bacteria is performing sexual reproduction. When the bacteria undergoes sexual reproduction by the process known as conjugation, at that time two sexually reproducing bacteria, they come close to each other. The pili forms a bridge which is known as sex pili, and through this bridge a segment of the DNA from one bacteria moves to another bacteria. So the bacteria which transfers its part of genetic material, that bacteria is known as gram positive, and the one which takes the bacterial DNA, that's known as the recipient. The one which gives is donor, and the one which receives is known as recipient bacteria. So this is the function of pili. Pili serves as the cytoplasmic bridge between the two bacterial cells for the transfer of genetic material from donor to the recipient bacteria. And the last one, but one thing you mind, that the pili is not a locomotory process like flagella. The function of flagella was it helps the bacteria to float, it helps the bacteria to move, but pili is meant for some other purpose. It is a non-locomotory structure. The last one is fimbriae. It is again a non-locomotory cell process which has no function to perform in locomotion. Rather, it helps the bacteria to stick to a surface. If bacteria attaches to our skin, the fimbriae of the bacteria, it helps it to stick to our skin. So these are the three cell processes which a bacteria has, and the three different functions you know now. Now what is cytoplasm? Cytoplasm is the part of plasma membrane. Actually, cytoplasm is the structure, it is a jelly, it is the matrix which is present, limited by the plasma membrane. And it actually is the living matter which is responsible for all the metabolic activities happening inside the bacterial cell. So this cytoplasm of the bacterial cell, it has got a specific region in the center. That specific dense region inside the cytoplasm of the bacterial cell is known as nucleoid region. What is special about the nucleoid region? The nucleoid region has got the genetic material inside it, DNA. So actually that's why this region is known as nucleoid region. Unlike the cells of we human beings or the plant cell or the fungal cell, the nucleus, the nucleoid region doesn't have a limiting membrane around it. That's why it is known as a nucleoid region, not a clear-cut nucleus. Okay, now let's see the next structure. You can see some twisted structures arising from the plasma membrane which project inside the cytoplasm. These coiled structures, they are known as mesosomes. So what are mesosomes? They are membranous structures found as an extensions or infoldings of plasma membrane which penetrate into the cell. Now these extensions, they are in form of vesicles. What are vesicles? Small sacs. They may be in form of tubes, tube-like structures known as tubules, or else they may be in form of elongated tubular structures known as lamella. Now what's the function of mesosome? The mesosome is responsible for formation or secretion of peptidoglycan, and hence it forms the cell wall. The second function of the mesosome is that it helps in DNA replication. And once the DNA has replicated, it distributes the replicated DNA into the daughter cells during division of a bacterial cell. The next function of mesosome is that it is actually like mitochondria of the eukaryotic cell. It is the site where respiratory enzymes are present. Hence, here the respiration happens, the food is oxidized, and energy is produced. So this is actually the powerhouse of a bacterial cell. This also helps in secretion of substances which happen from the bacterial cell. Then it helps in increasing the surface area of the plasma membrane. Now, in some of the prokaryotes like cyanobacteria, this mesosome, it helps in formation of those specialized structures, sac-like structures known as chromatophores, which contain pigments. And what is the function of the pigment? They trap the solar energy and they help in autotrophic nutrition of the cyanobacteria. So mesosome is a very good multitasker of a bacterial cell. It performs so many functions ranging from cell wall formation to DNA replication and distribution to respiration to secretion, then performing enzymatic activities by increasing the surface area of plasma membrane, and lastly it is responsible for synthesis of food in cyanobacterial cell. Now let us see the genetic material of the bacterial cell, which lies inside the nucleoid region of the cytoplasm. What is this genetic material like? The genetic material is a piece of double-stranded DNA which is made circular. It is actually a circular piece of DNA lying in the nucleoid region, but it is not limited by a nuclear membrane at all. This DNA is also known as chromosomal DNA or genomic DNA. And what's its function? Its function is transmission of characters from one generation to another generation of the bacteria. Next, there is another DNA also present in some bacterial cells. That DNA is actually not the DNA present in the chromosomal DNA. It is different, so it is also known as extra chromosomal DNA. This extra chromosomal DNA, its technical name is plasmid. This also is a circular double-stranded DNA, but it is quite small in comparison to the nucleoid DNA. And its function is that it has got genes to provide antibiotic resistance to bacteria. And that's why you would have heard certain bacteria, they become resistant to the antibiotics, and it is very difficult to fight with those diseases which are caused by drug-resistant bacteria. Now, the next is the ribosomes. So what is a ribosome? The ribosomes are small structures, granular structures present inside the cytoplasm, which are chemically ribonucleoprotein in nature. So this ribosomes are the structures which are not again surrounded by any membrane around them. The size of the ribosome is 15 nanometer. You can see how small its size is, 15 to 20 nanometers of size. And each ribosome of a prokaryotic cell is known as 70S ribosome. What is the 70S? S is actually Swedenborg's constant. This Swedenborg constant, it refers to the sedimentation coefficient of the prokaryotic ribosome. So the 70S ribosome of a prokaryotic cell, it has got two subunits. One subunit is the larger one, which is 50S subunit, and the other one is 30S, the smaller subunit of ribosome. So these two together, they form a 70S prokaryotic ribosome. And what's the function of ribosome? It is meant for protein synthesis. So while the ribosome is performing protein synthesis, what does it do? It attaches itself to the messenger RNA molecule, and there are several ribosomes attached to the messenger RNA molecule, forming a structure known as polysome or polyribosome. So now the last thing is the inclusion body. So there are certain reserve materials present inside the prokaryotic cell. These reserve food materials, they are known as inclusion bodies. These inclusion bodies, they are bound by certain membranes. Okay, now let us see what are the different inclusion bodies present in a bacterial cell. First is the phosphate granules. Phosphate gets stored inside a bacterial cell. There are some cyanophycean granules granules and certain glycogen granules present in a bacterial cell. Along with it, sometimes some gases also are enclosed inside the membrane, forming gas vacuoles inside a bacterial cell, which are meant to provide buoyancy, flotation to the bacterial cell. Now, this is the assignment for today. There are a few multiple choice questions which I have given here. Please try and solve it. Thank you.